Okay, so I think we can get started because we have a very interesting agenda for today. So good day to everyone from wherever you are seeing us. Welcome to the webinar from today. My name is 𝗥𝗲𝗱 𝗕𝗹𝗹𝗀𝗀. I am a Senior Marketing Associate at Nanion Technologies, and I will be your host for this session. So we're very happy to see so many of you joining us for this session as we get insights into the advances in automated patch clamp analysis of red blood cells. Before we get started with the presentation, I would like to give you some general information, some rules. So yeah, by default, your video is off and your audio is muted. So please try to keep it that way so that we ensure that we have a very nice flow in the session of today. As you probably noticed already, this webinar is being recorded and it will be later available to all of you. And also we will have a joint Q&A session, so at the end of both talks. So that should not stop you from writing your questions in the chat as they come into your head. So please feel free to write the name of the speaker and then your question. And at the end, we will open the discussion and read them to the speakers. Yeah, so they are addressed. Concerning the agenda, we will start with an introduction that will be given by my colleague Artem Kondratskyi. Later, we will move on to the talk from Nicoletta Murciano. And last but not least, we will have the research insights from Lars Kaestner. So I think we have a lot of interesting points to learn today, so I think we can get started. Yeah, so it is my pleasure then to leave you with my colleague Artem, who would be giving the introduction and would be also the moderator for today's session. So when you are ready, Artem, you can start sharing your screen.
Yes, thank you, 𝗥𝗲𝗱. So hello, everyone. It's wonderful to see so many specialists in red blood cells and ion channels joining us for this webinar. Today, we are going to talk about automated patch clamp and red blood cells, so to say APC and RBC. Yeah, as 𝗥𝗲𝗱 told us, we're going to have two talks today by Nicoletta Murciano and Lars Kaestner. But before this, we were thinking that a short introduction will not probably hurt. So let me do it for you right now. And I think I'm going to start with telling a few words about Nanion. I think that many of you know already about us, but if not, or maybe you forgot something, we at Nanion, we produce electrophysiology instrumentation for life sciences. You can see we have a bunch of instruments for various applications, and we like to categorize them into three groups: automated patch clamp instruments, membrane biophysics instruments that comprise transporter research instruments and lipid bilayer stations, for example. And also the third group is cell analytics instruments. So if you happen to work with cardiomyocytes, for example, we have very cool instruments that can measure cardiomyocyte contractility and viability. And if you are in cancer research, then our instruments can help you measure in real time cell viability, proliferation, adhesion, barrier function. So very, very nice instruments. But for today's webinar, we're going to focus on automated patch clamp. Here are the instruments that we propose. They have different throughput, from low throughput to the very high throughput. And of course, when we think about automated patch clamp, one of the first advantages that comes to mind is high throughput. And it's normal because these instruments are designed to help you obtain data more easily and rapidly than you can do with manual patch clamp, for example. But throughput is not the only advantage of this system. There are others, and one of them is that these systems can make much easier for you to patch small cells and organelles, for example. And here are three examples that I wanted to show you: data from red blood cells or mitoplasts or lysosomes that are recorded using planar automated patch clamp instruments. And again, since the webinar is about red blood cells, I wanted to tell you a few words about these cells. So due to their kind of minimalist structure, these cells are often teased by scientists. So here are a few nicknames that they got from scientists: a membrane without much of a cell, or an empty bag of hemoglobin, or hemoglobin sacks, or even cellular donuts. And all these nicknames, they can, give you an impression, a false impression, that these cells are kind of not very serious or not very important. But of course, it's not the case. Red blood cells are critically important for us, and they constitute about 70% of all the cells in the human body, which is actually insane. These cells also have been implicated in various diseases, and that's why it's not very surprising that researchers are interested in studying them. And what we see is that the RBC research is growing, publications per year, and researchers understood this very early, that RBCs are actually a very good model for studying membrane transport. And why is that? Because these cells, they have no nucleus, no organelles, so no contamination from inside. They are naturally in suspension, so we don't need any mechanical or enzymatic digestion for putting them in suspension, and they are very easily obtained from the blood. So obviously, we can hardly think and dream of a better model. And because of all this, several groundbreaking discoveries actually connected to red blood cells. And these are, for example, 𝗥𝗲𝗱. It was discovered in red blood cells for the first time by Peter Egger, and he got the Nobel Prize for this. Also, plasma membrane calcium ATPase or pump leak mechanism, all these were discovered for the first time in red blood cells. And later on, a variety of red blood cells transporters have been discovered on the membrane. And of course, we will not forget to tell you about ion channels in these cells. So for the moment, a few ion channels also have been discovered in these cells and characterized there, and also they have been linked to some physiological processes like mechanosensing, maturation, or cell volume regulation in these cells. And also for some pathophysiological, some mutations, they are connected to red blood cells related diseases. So looks like ion channels are very interesting targets to study in red blood cells, but let's look at this graph, and we'll see that actually red blood cells and ion channels research constitute just a tiny portion, tiny part of all this total RBC research. And if we magnify this red part and see closer here, we will see that actually there is no any growth in terms of number of publications per year. So if we compare here with the RBC research, which is growing, and then look here, no growth at all. And this is quite surprising because RBCs were one of the first cell type that was tested with patch clamp. Owen Hamel in 1981 recorded the first RBCs, but still no increase, no growth in research. Yeah, there should be some reasons for that. And one of the possible reasons is actually that patch clamping red blood cells can be a quite challenging exercise. And this is because of the small size of these cells. Their membrane are quite fragile and deformable. But even if you manage to patch them, you will have to deal with a limited copy number of channels per cell and high cell-to-cell heterogeneity. So it can be really stressful to patch clamp red blood cells. And we all know that stress is not good for our health. So we need to do something to eliminate this stress. And the obvious and logical question here is, what can we do about it? And this is exactly the topic that will be addressed in the first talk of today's webinar by our first speaker, Nicoletta Murciano. She is an application scientist at Nanion, and she has background in biology. And now she is also enrolled in the PhD program at Saarland University, where she's studying the role of ion channels in red blood cells in health and disease. Hopefully, Nicoletta will share with us how she deals with this stress and what approaches she's using to eliminate stress related to patch clamping RBCs. So over to you, Nicoletta, and you can start whenever you're ready.
Thank you, Artem, for this nice introduction. Today, I'm going to talk about the electrophysiological characterization of red blood cells using the automated patch clamp. I will start my presentation with a brief introduction of the role of calcium in red blood cells. So calcium is involved in basically in the entire life of red blood cells, starting from their production during the erythropoiesis process. Then is involved in the volume regulation of red blood cells. That is a very important requirement for the main function of red blood cells. So transporting oxygen through the body. Volume need to change to allow cells to squeeze through the narrowest capillaries. And also towards the final steps of red blood cell life. So during the aging and the clearance of red blood cell from circulation. Calcium is tightly controlled in red blood cells via membrane transport. This is allowed to keep a big difference in calcium concentration between the intracellular and extracellular environment. And this is mainly controlled actively by the plasma membrane calcium pump and by ion channels. When this tight control mechanism is impacted, the risk is to have a toxic overload of calcium. This causes the acceleration of red blood cells clearance, and this can lead to anemia. So for this reason, I decided to focus on ion channels critical for calcium regulation in red blood cells, and in particular on these three channels. So we have Gardus, that is a calcium-activated potassium channel, and its mutation is involved in the Gardus channelopathy. Then we have Piezo1, that is a mechanical activated non-selective cation channel and is connected to the hereditary xerocytosis. And the most recent discovered, at least compared to the other two channels, TRPV2 channels, discovered in red blood cells in 2021, and this nice study from 𝗥𝗲𝗕. And its role in physiological and pathophysiological function in red blood cell is still ongoing. So we all know that the manual patch clamp is the gold standard technique for studying ion channels. And we can see here briefly the setup for the manual patch clamp technique. So we have the cell that is in the extracellular solution. Then we have the pipette containing the intracellular solution that is positioned on the cell. And via a tight seal formation between the pipette and the patch of the membrane, we have the formation of the gigaseal. And then via suction, it's possible to break the membrane, get the whole cell access, and record ions passing through the hole. With the advent of the automation of this technique, so with the planar automated patch clamp, the main change was the replacement of the pipette with the chip. So now there is no pipette, but the cell is dispensed and positioned onto the hole. It's always in the extracellular solution. We have the intracellular solution above. And then via negative pressure, it's possible to break the membrane, so to get the whole cell access and to record ion fluxes from the whole membrane. So this process was scaled up in Nanion, reaching the high throughput APC system 𝗥𝗲𝗱 𝗕𝗹𝗹𝗀 that I use for my studies. And we can see a picture of the system here in total, and we can see the 384 chip. So the system allowed to record 384 cells in parallel. Each well can contain one hole minimum, but we have also chips with 4 holes and 8 holes per well. And then we can see the picture of how the deck of work look. So in principle, how it works. We have containers, so reservoirs, where we have solutions and cells that are aspirated and dispensed by an arm robot equipped of 384 tips inside the measurement chamber. So this is the measurement chamber where the chip is inserted and where all the patch clamp reaction takes place. And here we have the robot with the 384 tips that aspirate and dispense solutions. So which are the advantages of automated patch clamp for red blood cell study? So Artem already mentioned many assay challenges. Patching red blood cells is challenging with both technique, so it's quite stressful. because we have many challenges, like the low number of receptor copies. This leads to small currents. These cells are very small and high deformable, and being a primary cell line is difficult in revealing channel identities. But let's look at some advantages from the APC. So obviously, having 384 red blood cells recorded in parallel, it helps to collect a big amount of data in a shorter and easier time compared to manual patch clamp. But this helps with, in particular, to test the heterogeneity of naive red blood cells conductance. And it helps also to detect functional phenotype, for example, when we want to study an unknown mutation of the channel compared to a control, for example. Now I will move on the study objectives. So as I said, I was focusing on these 3 channels, and in particular, I wanted to study piezo using the specific chemical activator 𝗲1 during differentiation. So I studied piezo in erythroblast, reticulocytes, and mature red blood cells. Then I wanted to combine the chemical stimulation with the more physiological activation, so with mechanical stimulation. And I did perform these experiments in erythroblasts and mature red blood cells. And then I will show some results, this study of a patient with an unknown mutation of piezo investigated with 𝗲1. Then I will move on TRPV2 channels. TRPV2 channels are activated by delta 9 THC, and we will see the behavior of the channel in healthy donors versus sickle cell anemia patients. So I would like to specify that this study was done using different techniques, but the patch clamp part was done with manual patch clamp. But I put here my paper, so our collaboration, where we were studying TRPV2 channels using automated patch clamp, testing healthy donors, but between smoker and non-marijuana smokers. Then Gardos was parallelly investigated with these 2 channels. Because since the activation of piezo and TRPV2 led to calcium influx, these activate Gardos. So what I did was to apply some Gardos blocker, TRAM34 and caryptotoxin, to see how the channels interplaying among us in the different study cases. So moving on the first result section, I'm starting with the characterization of piezo activity in cultured erythroblast, cultured reticulocytes, and mature red blood cells. So the cultured erythroblasts and reticulocytes were provided by the blood bank Sanquin in Amsterdam. And so moving on the graph, we can see in A and B some representative traces and current trend from erythroblast. Starting from panel A, we can see current over time over different additions, with in gray the baseline, so the current before the compound, before 𝗲1. Then we can see the jump of activation after the addition of 10 micromolar 𝗲1, and then the block via 30 micromolar gadolinium chloride. Here we can see the ramp traces, because the voltage protocol applied was ramp ranging from -100 millivolts to +80 millivolts. Same color legend, so pre-compound 𝗲1 and gadolinium chloride as blocker. Then for the statistical analysis, I've looked at 2 parameters in the 3 samples. And I was focused only on the positive voltages, so only on the +80 millivolts in the ramp. And you can see, so the compound-induced current for 𝗲1 and 𝗲1 plus TRAM34, and the percentage of responding cells over differentiation. So we can see here in light blue that 𝗲1 was able to activate piezo in the 3 cell types. With a slight increase, so with a slight higher current in red blood cell, even if not significant. When I did apply 𝗲 plus TRAM34 just to see the participation of Gardos to this component to the 𝗲 current, I did observe a significant decrease only in erythroblast currents, slight decrease in reticulocytes, and no decrease, but actually a slight increase in red blood cells. While for percentage of responders, the trend was quite clear. So there was significantly decrease over differentiation, reaching the lowest value in red blood cells. We are close to 10% responding cells. So it's almost 1/3 compared to reticulocytes and erythroblasts. Then I activate the channel. I've tried to activate the channel mechanically in erythroblast and red blood cells. Here is a brief introduction on how we perform mechanical stimulation. So this is the well view from the side one of our well of the chip. You can see the pipette that is entering the well, and mechanical stimulation is achieved by fast application at fast speed, 110 microliters per second, of 20 microliters solution volume on top of the cell, and then followed by a fast removal. This mechanical stimulation results in the inward current from piezo 1. And so I did perform this assay on erythroblast and red blood cells. In dark blue, we can see the pure mechanical stimulation, while in light blue, we have mechanical stimulation combined with 𝗲1. And we can see that in erythroblast, the mechanical stimulation elicited the small current, and this was amplified and boosted by the help of 𝗲1. But this was not possible in red blood cells. And if we look at the statistics of the percentage of responders, we can see the clear difference in the responding cells. And if we look at numbers here, basically only one cell on most 300 cells in total was able to reply to mechanical stimulation in the red blood cells, mature red blood cells population. So brief summary of this first part of results. Piezo was activated by 𝗲1 in erythroblasts, reticulocytes, and mature red blood cells. There was a significant decrease in current observed in erythroblasts with the Gardos channel blocker TRAM34. Percentage of 𝗲1 and 𝗲1 TRAM34 responding cells significantly decreased over differentiation, and piezo was mechanically activated only in erythroblasts. So here some explanations of why we observed these results. So during the terminal erythroid differentiation, many things happen. So as we said before, there is the decrease of copy number of the low copy number of channel, so it's decreasing over differentiation. Then through the differentiation process, there is a big loss of the composition of the cytoplasm and loss of membrane, so also the possibility of the loss of protein inside the membrane. Then we have to consider that the opening of the channel can be random. Low number of copy plus random opening of the channel can lead also to have a low percentage of responding cells. And then there is a severe lipid remodeling associated with the aging process. And since we know that piezo is highly influenced by the lipid environment, this can also explain why our setup for mechanical stimulation was not able to activate red blood cells. So now we'll move on the next result section. So we have family case. We have the father with the spectrin, alpha spectrin mutation, mother with the piezo1 mutation, and the patient, so the daughter with both mutation and clinical manifestation, while the parents do not have any clinical symptoms. So we have severe hemolysis and stomatocytosis, both suggestive of hereditary 𝗥𝗲𝗃. So I received the samples before splenectomy. So the question was if the function of piezo could be related to having this mutation, could be related with the hereditary 𝗥𝗲𝗃. I received the sample before splenectomy, then the patient underwent splenectomy, and I did repeat the analysis. So here we can see the results before splenectomy. I looked at again at 𝗲 induced current, percentage of 𝗲 responding cells, and baseline current. So basically the current before the application of 𝗲1. The iode-induced current was, I could not see any difference in some whole samples. We have also further an additional control from a healthy donor. But what was clear was the difference in percentage of responding cells. So we can see that the patient, the daughter, is around 1%. And this is significantly lower compared to the other samples. And I put also a representative cell on how the current trend looked over addition. And you can see that it's more run down, and the addition of iode doesn't show any activation. I can also add that the seal over time was decreasing. So I would say that patch performance was very low for the status of this of the patient cells. And the baseline current, so before the iode addition, was significantly higher compared to the other samples. And this is something that was also seen in cases of hereditary 𝗥𝗲𝗕𝗹𝗹𝗀, so cation imbalance in the baseline. Then we have the results after splenectomy. The iode-induced current looked like so before, no differences, but I could see immediately an improvement in the percentage of responding cells and also in the current trends. Was closer to the control and the other samples, and same for the baseline current was close to the healthy control. So here another two graphs just to show the impact of splenectomy on iode-1 induced responses. We can see putting directly only the daughter results before and after splenectomy, there is a significant increase in the percentage of responding cells. So in conclusion, splenectomy led to improvements in the patient red blood cells population, enhancing piezo activity and bringing red blood cells behavior closer to that of healthy controls. Then we have the part regarding TRPV2 activity in red blood cells of sickle cell patients. So first of all, I would like to explain why we decided to study TRPV2. And sickle cell anemia is a genetic blood disorder that is characterized by a mutation in the beta globin subunit of hemoglobin, resulting in this hemoglobin S. And in the deoxygenation state, we have this typical cycling of the cell, so that the hemoglobin polymerize leading to this cell shape. Then these cells are characterized by an increased cation conductance that can lead to the Gardus effect. So having calcium entering the cell will activate Gardus that will cause the efflux of potassium, followed by chloride, and then osmotically by water, causing severe dehydration. So since TRPV2 is allow the influx of calcium inside the cell, we wanted to see how this impact sickle cells applying THC, so activating TRPV2, since these cells are even more sensitive to calcium changes. We performed this study in collaboration with other laboratories. We had a group of healthy donors, a group of sickle cell patients, and we decided to perform different techniques. So flow cytometry, manual patch clamp, and the MBE method. Starting from the manual patch clamp results, so we can see the AA is the control as we have the sickle cell patients, and we can see the ramp protocol that was applied was ranging from -100 to +100 millivolts. And we can see row traces before and after THC addition, 30 micromolar THC. We have an increase in both samples, but when we look at the membrane conductance and in positive voltages, we could see that the increase in conductance after THC application was significant only in the patients and not in the healthy control. Plus, the percentage of responding cells was more than the double in the patient, with 47% against 20% in healthy control. So we decided to look at the MB method. So at the MB method, you can record the changing in the membrane potential from the whole population of cells. And here we can see how the graph from MB technique look. And we have the voltages against time. And we can see how the potential change with the addition of THC. In orange, we have the healthy control. In blue, we have the patient. And we can see that with the application of THC, the hyperpolarization that is due to Gardner's activation, because Gardner's with the potassium efflux make the membrane more negative. So we can see that in the patient, the hyperpolarization is bigger. And we did perform the technique in oxy and deoxy condition. And the control is not impacted by the deoxy state, but in the sickle cell, we can see further increase of the hyperpolarization. Then when we look at the statistics, we can see that this is significant only in the patient, and also we can see the application of 𝗥𝗲𝗕𝗹𝗹𝗀. 𝗥𝗲𝗕𝗹𝗃 is a Gardner's blocker, because we wanted to confirm that this is the Gardner's effect. And in fact, we can see that with the addition of 𝗥𝗲𝗕𝗃 in oxy and deoxy condition, the membrane potential is going back to normal values, so similar to the healthy control. So since Gardner's is activated by calcium, we wanted to look at the calcium movement inside these two samples. And that's why we perform flow cytometry using the fluorophore market, and we look at the fluorescence intensity. And so in the graph G, we can see that already at the resting state, sickle cell red blood cells have a significant higher calcium content than the healthy control. But that it's something that we expected. But the number of responding red blood cells and intensity of fluorescence was significantly higher in sickle cell patients after THC addition, 80 macromolar THC addition. And so we are around 25% against 50%. So this is aligning with the patch clamp recording. So in conclusion, red blood cells from sickle cell disease patients are more responsive to acute THC stimulation compared with control red blood cells, without any difference in the copy number of TRPV2 channels. I forgot to say this, we perform Western blot without seeing any difference in the expression of the channel. So we can explain the results saying that in sickle cell, having a higher calcium level, the threshold for Gardner's activation is lower. So that's why the channel activation lead to faster and higher Gardner's response. And since cannabinoids derivatives are used for pain treatment in this patient, this should be considered more with caution, and would be interesting to try to establish a correlation between smoking cannabis and the severity of sickle cell disease for smoking patients. Yeah, this was my last slide. We'd like to thank all the collaborators in these studies, and of course, thank you all for listening.
Good, very nice. Thanks a lot, Nicoletta, for this presentation.
Yeah, thank you.
And I hope our audience has a lot of questions to you. I certainly do, but let's maybe have all this Q&A session after the presentation of Lars. Yeah, so our next speaker, ladies and gentlemen, is Lars Kaestner, professor at Saarland University. He has a very interesting background. Initially trained as a physicist, he then specialized in biophysics and became increasingly involved in medical topics. His PhD studies are directly related to electrophysiology and biophysics of red blood cells. He is initiator and coordinator of a few international training networks related to red blood cells, like Evidence and Relevance. He's also an organizer of multiple international conferences on red blood cells and has experience with industry too. I'm almost sure that most of you know Lars very well, but if not, I wanted to show you just this image of collaboration network in ion channels and RBCs field. Here you see the authors that work in this field, and the bigger the name, the bigger the contribution of the author to the field. So, ladies and gentlemen, please welcome one of the top voices from the field of ion channels and RBCs now, is Lars Kaestner.
Well, thank you very much for the kind introduction. As you see, we have a very well-tuned introduction. So I even matched today the shirt I had on the introduction photo. Well, I'm happy to present once more in this webinar format, and I'm happy to see lots of old friends in the participants, but also lots of people I don't know. So well, anyway, I try my best to give an interesting presentation. Okay, well, I'll talk about pseudo action potentials in red blood cells today, and I'm sure most of you never heard about pseudo action potentials because, well, I just came up with this term last year, so it's normal you don't know it, but I hope after the presentation you will be convinced about the concept. So, well, I continue with Artem's nickname slide, but put it maybe on a more scientific base. So I think cell can't be more of a prototype of a non-excitable cell than a red blood cell, because well, we have lots of things that are typical for an excitable cell. I mean, the reservoirs, the way of the calcium signaling, the action potentials, all that is absent in red blood cells. But what is special about red blood cells in electrophysiology? Well, one major property that is completely different to all other cells is the extremely low membrane permeability for cations, and in particular for potassium, at least in the resting state. And this means, in turn, that the resting membrane potential is not determined by the potassium, as in most other cells, it's determined by the chloride. And this means the resting membrane potential is approximately minus 12 millivolts. And, well, what is also special about red blood cells is what we already heard before, that the copy number of ion channels is very low. I mean, for a particular type of ion channels, it's well, most probably for all channels below 100, for some even lower. And, well, what you also always need to keep in mind, there is no translation activity. So whatever the cell has, or most of the things the cell has, needs to be there from the very beginning. And, well, I also try to keep my presentation simple. So I will mainly talk about 2 ion channels only, with a very few exceptions where I make reference to other channels. And I'd like to start with the evidence for the CAF 2.1. In the previous slide, we have seen a big question mark, or even several question marks behind this channel. And indeed, it's a kind of a phantom channel in red blood cells. So what I show you here in the first panel is a Western blot of the alpha 1A subunit of this channel, and this is the area where you need to look at. So this is the Western blot band of three different donors, and in A you see the normal Western blot, and in the B lines you see how it looks like if well, if the proteins were incubated with a shedding peptide, so that is here used as a proof of the specificity of the antibody. And then what we also have in large amounts in several studies, that we have an inhibitor of that channel, what is called 𝗥𝗲𝗱 TK. And well, we have a number of signaling cascades in the red blood cell where we believe, or where I believe, this 𝗥𝗲 2.1 is involved. And if we apply this omega-agar toxin TK, we can inhibit this this function. Well, as I said before, it remains a phantom because I personally never have consciously seen any 𝗥𝗲 recording, so any direct channel openings of this channel during my entire scientific life. On the other hand, if we look at these calcium imaging experiments, as you see it on the right, this is a method where signal is integrated. So even if you have very small signals that you may not be able to see, you can over time integrate the function, and then you see well, even significant differences. So what is this all good for? Because the 𝗥𝗲 2.1 is, as the name already indicates, a voltage activated calcium channel. Since I just told you the red blood cell is a prototype of a non-excitable cell, well, why a voltage activated channel should make sense. And well, with my next slides, I come to the second channel, and this was already mentioned. I just want to take the time to take you to the natural history of that channel. What is the so-called Goddard's channel? Well, initially reported as the Goddard's effect by Goddard's in 1958, and well, if you look for the original paper, it's something like a one-page paper. So, well, this is a hint for the young scientists. So if you do the right things, one page of paper might be enough to have your name in science forever. So this is then, to my best knowledge, the first official recording of the first official 𝗥𝗲 recording of the Goddard's channel, presented at the meeting of the Physiological Society in 1981 by Haenel. So this was a poster, actually, not a talk. And well, only in 2003 we got the first paper by Joe Hoffman that gave well, let's say, a very strong indication that the Goddard's channel is indeed the HSK4 or the KCNN4, depending what nomenclature you follow. So this was the first, let's say, the first written concept where the Goddard's channel was dedicated to a molecular identity. And I think this was finally confirmed and without doubt when there was mutations of the Goddard's channel related to red blood cell phenotypes in hereditary diseases. So this was one of the first reports in 2015. And at that time, I think it was still called hereditary or dehydrated stomatocytosis. I think in the meantime, we know that the dehydrated stomatocytosis is mainly related to mutations in the piezo channel, and the gain of function mutations in the Goddard's channel are associated with the pathology we call Goddard's channelopathy. Okay, so far about the history of the Goddard's channel. I now come to a recent publication from this year, and Nicoletta also referenced this paper several times. And one panel of this paper shows a proteomic analysis of some of the ion channels, and it shows that even state-of-the-art proteomics is not able to reliably determine the Goddard's channel, neither in reticulocytes nor in mature erythrocytes. And well, I can say that this proteomic analysis is done by a core facility of St. Queen in Amsterdam. So, I mean, these guys, they measure red blood cells, well, every other day. They have the latest technology and equipment. So it's not any kind of village laboratory that is not able to detect this. It's really the state-of-the-art laboratories. I mean, from time to time, one can see it, but not consistently have it in all samples. So what we can really say is that the Goddess channel is at the detection limit of modern proteomic analysis. Well, and this is obviously related to the very low copy number of the channels. And well, we have the remaining discussions for the CAF 2.1 that I already mentioned, and a similar discussion for the 𝗕𝗪C6 that, well, could never be really shown in Western plots to be present. But again, functionally, we see something like 𝗕𝗪C6, but this is only a side notice. And what you also need to keep in mind that if the protein is present in the red blood cells, and it means you may detect it by proteomic or other molecular biology analysis, it does not mean it's still functional. Because as I said before, and as we all know, there's no translational activity, and in principle, there is no renewal within the 120 days average lifetime of the red blood cells. Okay, so now the question is, what happens with the membrane potential when the Goddess channel is activated? And what I have plotted here is a diagram, membrane potential against time. And what you see on the very left is a small black line. What is the resting membrane potential at -12 millivolts? And the method used here is the method Nicoletta also referred to already. It's called the MBE method to measure the average membrane potential in red blood cell suspension. And well, I decided to mention here the core, well, the boundary conditions for this method. I don't expect, if you never heard or familiarized with this method, to understand exactly what's happening. It also took me several years to roughly understand that. It's based on a protonophore that is given to the red blood cell, the CCCP. And this allows the protons to freely move across the membrane. And another essential part of the method is that you use a pH unbuffered external solution. And on the other side of the membrane, you have the hemoglobin, which is a good proton buffer. So this means the internal pH value is clamped due to this buffering capacity of the hemoglobin. And as a result, now the protons will distribute according to the membrane potential. And therefore, the external pH value allows you the determination of the membrane potential. Okay. That's the method. You probably need to think about a little bit about this. As I said, you don't need to understand the method to understand the graph. And here you see what happens if we activate the Goddess channel with a compound called NS309. Well, it's not directly an activator. We will see it later. It's just, it reduces or increases the calcium sensitivity for the Goddess channel, so it opens up easier. But for now, let's look at the graph as it would be an activator. And what you see is that in the entire cell population, the membrane potential drops from -12 to below -50 millivolts. And well, what you can see there is indeed red blood cells at least can polarize. Yeah. And well, if we continue the experiment, if we add a Goddess channel blocker from 34, we can inhibit this response, and then usually at this end of these measurements, we lyse the cells with Triton X 100, what gives you the calibration point of 0 millivolts. So, I mean, this is obviously not yet what I refer to as pseudo action potentials, because we see the polarization, but except if we add the TRAM 34, there's no depolarization. What would be kind of the characteristics of a membrane potential, or of the action potential, sorry. And in addition, I could not think of any physiological stimulation that, well, maybe for switching on the Gauss channel for the polarizing, I could think of a physiological signal, but to switch it off in a synchronous way, I can't think of. Yeah. So the solution to what I call the pseudo action potential then comes in the next slide. And well, I like to remind you once more the low Gauss channel copy numbers per cell. Nobody knows how low it really is. I have 3 references that did an estimation of the channel numbers. So, well, in the 𝗥𝗲𝗱 et al. Paper, they compared channel activity with sodium or rubidium fluxes through the Gauss channel. Wolf et al. A few years later estimated 2.6 channels per cell. In our paper this year, by comparing whole cell patch clamp with single channel conductance, we came to something like 3.1 channels per cell. So the same order of magnitude. Of course, this is functional channels because we measure patch clamp. So the protein number might be bigger, and it's also an average value. So the particular cell may have a little more or even less. And now I think I show the only internal patch clamp measurements that were indeed recorded with a patch liner. And these are whole cell Gauss channel recordings. And what you see here is these recordings almost look like single channel recordings. Yeah? And this is not so surprising if you consider the channel numbers per cell. And if you have these channel openings on the base of a single cell, then this obviously also induces these membrane potential jumps. And the idea is that if you have a flickering of the Gauss channel, you have this polarization and depolarization cycles. So now the question that comes up is, do I have any proof that these pseudo action potentials indeed activate this CAF channel? Because I mean, there are many kind of small details that could argue against the big ideas. What I can do is I can show you some indications, but I don't have real convincing patch clamp recordings. So I'd like to go or take you back to these membrane potential measurements, and we did exactly the same measurements in different concentrations of calcium. And as I said, the NS309 increases the sensitivity for the calcium. This means if you have a low calcium, we have a low average change in membrane potential, and with increasing calcium concentration by maintaining the NS309 concentration constant, we get an increase in the amount of average polarization of the red blood cells. So if we have these polarizations and possibly depolarizations, we would also expect an increase of the calcium in the red blood cells. And as I mentioned before, the calcium is this integrating parameter. So let's have a look what the calcium does. And what is presented here is the numbers or the percentage of cells with a high calcium. And well, this is everything else than a linear relationship, but with having this this CAF 2.1 and the Goddess channel activity in background, it makes a lot of sense. So if you have a very low Goddess channel activity, so let's say at the 1 micromolar calcium concentration, then the Goddess channel is not very active. It's closed. You don't get much of a polarization or depolarization, and the number of high calcium cells is rather low. Then, if we increase the calcium concentration, then the Goddess channel becomes more active. It shows this stochastic behavior of openings and closings, what is compatible with significantly increased number of high calcium cells. But then if we then increase the open probability of the Goddess channel further, meaning the Goddess channel is open, this means there's no more flickering because it's constantly open, and this would mean less activity of the CAF 0.1 and hence also less responding cells with high calcium. So now the next question is this anything good for, or is there a link to real life? And here I'd like to take you to the situation where we look of patients with this Goddess channelopathy, meaning with a gain of function mutation in the Goddess channel. And to our surprise, well, just once more, it's a gain of function mutation in the Goddess channel, and the Goddess channel is a potassium channel. Yeah. Nevertheless, we see in the red blood cells of these patients an increased basal calcium, and we see this in microscopic images, but also in flow cytometry. And well, if you now look at the patient cells, if we incubate the cells for 24 hours with particular channel modulators, well, this is the result. So the left column, we have the control conditions, how the cells look like after 24 hours of storage. So we have a certain percentage of high calcium cells. If we incubate the cells with the Goddess channel inhibitor TRAM34, this number of cells is much lower, and exactly the same results if you use a different channel inhibitor, the 𝗕𝗏𝗃. Also, if we try to inhibit this CAF 2.1 with this omega-agadoxin TK, we also get a much lower percentage. But if we activate, or if we inhibit the piezo channel, piezo1 with GSMTX4, we get a higher amount of responding cells. What on the first view looks very paradox. Yeah, but in my summary, I will give you also the explanation for this. So here we have the red blood cell with the Goddess channel. If the Goddess channel is activated, either in the patients with the gain of function mutation, what increases the open probability, or experimentally with the application of the NS309, we get potassium loss. At the same time, we can manipulate this with inhibitors. For the Goddess channel, we have a bunch of inhibitors. Characterization is one. We have seen experiments with the TRAM34 or the 𝗕𝗏𝗃. Well, according to this presentation, with the pseudo action potentials, the activity of this Goddess channel can also activate the CAF 2.1. Well, if you reach this state of this membrane potential flickering, and with this, calcium can enter the cell and kind of also provide a feedback loop of further Goddess channel openings. But this would be also a kind of self-inhibition, as we have seen, because if the Goddess channel remains open, then also the CAF 2.1 channel would not open further. Also here we have the pharmacological tool, the omega-agadoxin TK, as we have seen. Well, the volume change may or may not, or there might be other reasons that activate the piezo channel. And the piezo channel, as we heard from Nicoletta, is a non-selective cation channel. Probably most of you also know the channel otherwise. Also here we have a number of different manipulators. So the 𝗕𝗲1 for activation and the GSMTX4 for inhibition are just examples. But at the same time, when this channel is active or open, it screws up the membrane potential, because it's equally conducting sodium and potassium. So it brings us far away from any attempt for polarizing the cell. So that means activity of piezo1 would kind of block or prevent the activity of the 𝗏2.1 channel. Okay, this was my summary. So the last slide I have is the conclusion. And my conclusion will not be a surprise for people like Anna or Stefan or the other friends from Roscoff. My conclusion is red blood cells are more interesting. They may appear at the first glance. And with this, I'd like to thank you for your attention. And I'm happy to discuss questions.
Yeah, thank you, Lars. Intriguing talk about action potentials in non-excitable cells. It was very interesting to listen to you. And let's see if we have any questions from the audience. Michael Netzer. So the first question will be to Nicoletta. Thank you for the great presentation, Nicoletta. I have several questions. Why did you not see a TREM34 response in RBCs on slide 7?
So yeah, slide 7, yeah, it's where I show basically the difference between 𝗕𝗲1 and 𝗕𝗲1 plus TREM34 in the different samples, erythroblasts, reticulocytes, and red blood cells. So yeah, I would expect TREM34 blocked in red blood cell as well. But yeah, as I speculate, this can be related with the decrease of copy number of the channel. So thinking about, as Lars said, having less than 5 copies per cell, not sure if it's 100% functional, because we don't have to forget that red blood cell with the aging, they lack protein translation. So probably channels can be also not functional anymore. So maybe is that the case that Cardos was not really activated with the influx of calcium via piezo and via 𝗕𝗲1. So this can be one of the explanations.
Another question, always from Michael Netzer. What was the current density for Gardus channels in erythroblasts?
Current density, meaning normalization of current with the capacitance. So what I've shown is just absolute currents. They are not current density. What I can say about the size of the cells, erythroblasts that were smaller than what we expected. They were very similar to red blood cell size. And that's why I use always also the same chip, same whole size for the all experiments. So that's why I didn't perform current density, so any normalization. And Gardos was only the results of the blocker addition. So I didn't investigate Gardos activation directly. So no current density.
And the third question from Michael Netzer, seeing the experiments with sickle cell RBCs, do you think there is a potential for a current clamp assay to measure Gardus channel inhibition?
Meaning so that the hyperpolarization with the MB method, I guess. So when we see that change in the membrane potential, I don't know, so I never tried. I don't know if red blood cells are suitable for this kind of experiment. Thinking also that MB method is applied on a whole red blood cell population. So many cells replying at the same time, we can see the change in the membrane voltage. I don't know, it's something that I can try on a single cell recording.
Another question is from Stefan Auger. Is it possible that the increased protein density in the membrane between erythroblasts and mature RBCs is also a reason why piezo-1 is already in hypertensive state and therefore less responsive in page clamp compared to other techniques.
So basically more proteins in the membrane of erythroblasts compared to red blood cells. And then is also the reason why piezo-1 is already in hypertensive state and therefore less responsive in patch clamp compared to other techniques. So what I can say here about tension of the membrane. So one of the reasons that I was attributing the non-responding of red blood cells to M-STEAM, for example, is the tension of the membrane of red blood cells in the whole cell configuration in the planar patch clamp. So my hypothesis can be that due to the high deformability of the cell is trapped inside the hole, and so the membrane exposed to the mechanical stimulus can be not at a good tension. So piezo cannot reply to the mechanical stimulus also for this reason, and also the exposed portion is very small. And about erythroblast, so my hypothesis is that we have just more proteins replying better to the mechanical stimulus, and compared to other techniques, of course. So this all cell configuration, the tension of the membrane can have an impact compared to other techniques, like the walking.
Well, I think 𝗕𝗹𝗪 is the person on this planet that knows most about ion channels and red blood cells. So the question is if Stefan keeps this or regards this as a possible concept, well, he should take it serious.
Yeah.
Okay, we have a question from Tom. With a resting membrane potential of -20 millivolts, it's a question to Lars. Would CAF 2.1 not be fully inactivated?
Yes.
I'm not familiar with its biophysics, and would it recover from inactivation when Gardas polarizes the membrane?
Yeah, exactly. This is the idea. Yeah. I mean, there is a number of things that are not clear or not 100% fit into the picture if you come from classical excitable cells, no matter whether these are neurons or muscle cells. So for instance, the amount of the depolarization is less as the resting membrane potential, for instance, in a neuron or cardiac myocyte. And I mean, you can think a lot about the efficiency or how action potentials are designed. And I mean, if you look again, either neurons or muscle cells, you know that there is 10 different channels contributing to the formation of an action potential, and you have this what I call the three-pool system. Yeah, you have the external side, you have the intracellular part, and in addition, you have the calcium reservoir in the endoplasmic or sarcoplasmic reticulum, and you need the tuning of all these three components to create the action potentials. All this we obviously don't have in the red blood cells. Yeah, but if you think about whether you could also kind of realize the same effects with a two-pool system in the red cells, just well, cytoplasm and external, it's possible, but it can be such nicely kind of synchronized. And what I also like to mention is we see it, for instance, in other channels that we do see differences in the channel properties. Let's say the sensitivity for drugs that are different in red blood cells than in other cells. So I could imagine even if we have a discrepancy of, let's say, 15 millivolts for the polarization that you know from excitable cell, it could still be possible by modulators in the membrane, the lipid composition, lipid rafts, whatever. I think there's lots of things we don't know yet.
Okay, thank you. And a question from Anna Bogdanova to Lars. Would it make sense to monitor calcium oscillations as readouts of membrane potential transients using fluo4?
Yeah, why not? I mean, I think the calcium oscillations Anna refers to are calcium oscillations that appear when you place red blood cells from a calcium-free surrounding in a calcium-containing medium. Then suddenly you see some calcium flashes and maybe oscillations. I think this is still not yet really explored.
Thank you. And I see Michael Netzer just add a comment. Thanks for the answers, Nicoletta. I actually was curious if the absolute Gardus channels, Gardus currents were bigger in erythroblasts versus red blood cells.
So the absolute Gardus current actually was not shown. So it's more like 𝗲1 elicited piezo responding, and then the block of Gardus with TRAM34. And no, so applying TRAM34, the current was smaller in erythroblast, so more channels were apparently blocked than red blood cells.
No, but if you isolate the Gardus current, it's indeed bigger in erythroblast than in.
Yeah, if you think about the opposite, so yeah, so I was not showing absolute current for Gardus. I want just to make that clear. But so if you think the portion of current that was blocked by TRAM34, it's true. So the component of Gardus current is bigger in erythroblast than red blood cells. Yes. But I didn't show that in my graph directly.
Okay, thanks a lot. I will probably ask one question from myself, very interesting about this action potential. So, Lars, have you ever heard about this phenomenon like pseudo action potentials in other non-excitable cells? I understand that it's kind of related to Gardus stochastic behavior, but maybe we can think of it in some other cells.
Well, I don't know. Yeah, I mean, as I said, this effect that we see, this activity or functional activity, or we see cellular activity that is compatible with the activity of this CAF 2.1. I think we follow this over a couple of years already. And I think just last year I came up with the term pseudo action potentials, and I haven't heard this in any other cells, and I would doubt that other cells have it, because this low copy number of the cells, what is essential for this flickering, I think it does not occur in cells that have a translational activity. So I think this could be something very special about red blood cells. Well, maybe similar on platelets. I'm not a specialist in platelets, but they have in principle a similar situation. They lack the translation and also have, for instance, 𝗥𝗲𝗈 in the membrane, so they also contain ion channels. So maybe there it's similar.
Okay, thanks a lot. That was very nice and interesting talk from both Nicoletta and Lars. We learned a lot. 𝗥𝗲𝗓, do you like to add anything in the end?
Yep, just for all of you that are still here, yeah, thank you for joining us today. And yeah, also we would like to extend the invitation for October 22nd and 23rd. We have our Nanion user meeting. So this is a hybrid event, and it will happen, so you can come in person or online. So yeah, so wherever you are seeing us today, you can just go to our website, register there, and you can also find a space, virtual space to join and see the different talks that we bring for you. So yeah, that would be all. So thank you also from my side, Nicoletta and Lars. And yeah, I hope other discussions and other webinars that we will have. Thank you then.
Thank you.
Thank you. Bye.
Bye-bye.
Thank you. Bye.